Hello, and welcome to the next in our OSTR series on cutting-edge methodologies. Today we're going to talk about structural mass spectrometry approaches, and specifically hydrogen-deuterium exchange mass spec. Hydrogen-deuterium exchange is a methodology for looking at changes in protein flexibility and protein conformation. In the method, regions with secondary structure, such as helices or sheets, tends to have low levels of exchange, whereas loops tend to have a higher level of exchange. And so by incubating proteins with deuterium, there will be an exchange between the proton and deuterium, and we can use mass spec to try and look at the difference in uptake of deuterium for protons. If you look at a single peptide like this, there are several hydrogens that could exchange. Within the side chain, there tends to be a very rapid exchange rate that goes so quickly that it's impossible for us to detect. In contrast, the aliphatic hydrogen on the carbon doesn't exchange at all, so that one we're also going to lose. Conveniently, the backbone amide group does have an exchange rate on the order of milliseconds to days, and so it's these exchange reactions that we're going to be analyzing. Hydrogen-deuterium exchange can be applied to several different biological questions. For example, one can ask, does a protein fold correctly? So the difference between different states and different folding forms can be identified by looking at changes in deuterium uptake. One can also look, are there conformational changes after application of a stress, after addition of a ligand, or changes in some form of the composition of the buffer or physiological condition. One can also ask, where does one protein bind another? Theoretically, in the binding site, there will be changes in deuterium uptake that can be used to map binding sites. One can ask, can I identify sites of protein aggregation, which would also affect deuterium uptake? One can look at how mutations affect protein structure, as well as where drugs interact with target proteins. Really, HDX is a nice methodology to look at protein structural analysis that enables the study of higher order structure, especially conformational changes and protein binding interactions. There are several factors that actually affect the HDX itself. These include solvent accessibility and hydrogen bonding, both of which are factors that report on the structure and conformation of the protein. Also, pH and temperature will affect the deuterium uptake, and these can be experimentally controlled. A general HDX mass spec workflow would begin with a protein alone or a protein plus a ligand. This is then incubated in D2O for a labeling period during which accessible protons will exchange to deuterium. Next is a very critical denaturation and quenching step at which the protein is kept at 0 degrees and an acidic low pH around 2.5. These steps are critical to maintain the deuterium that's on the protein and not allow back exchange to a proton. After this quenching step, proteolysis occurs using pepsin. Pepsin and other acidic proteases are the enzymes of choice for this type of analysis because they are functional at low temperature and low pH. After this proteolysis step, peptides are separated and then analyzed by mass spectrometry. These analyses can then occur over multiple different time frames such that the deuterium uptake over time can be analyzed, and one can look at changes in deuterium chain level in the protein over time. In addition, liquid handling steps are often used in an automated way to improve reproducibility and maximize instrument throughput so that more samples can be analyzed and there's less instrument downtime. There are several programs that are used for HDX analysis, and these are all shown on the screen. Some of these are publicly available and some of them are fee-based products, and they all have different benefits and disadvantages, so these are here for you as a resource. All of these methods, though, will go through the same type of step, which is acquiring data, defining what the protein is, looking at the mass spec data, detecting unlabeled peptides, and then using those to generate a HDX peptide list from the full experiment, detecting those modified peptides, and then interpreting the data and forming comparisons such that one can start to look at the effects of whatever treatment is being used. This is an example of a deuterium uptake map for a specific protein. You can see the sequence as well as different peptides that have been identified, And those peptides are color-coded by how much deuterium has been observed to be taken up across different times. The deuterium uptake is based upon mass shifts due to the change in mass from deuterium to proton. As you can see throughout the protein, there are different regions with either lower or higher relative deuterium uptake. Those different peptides can then be analyzed across different times, and one can see changes in the deuterium uptake at different times. In a standard experiment, there is some scrambling of the deuterium across the peptide such that it's hard to have single amino acid resolution. Methods using ETD for fragmentation do tend to maintain the localization of the deuterium on the residue. In addition, one can look at peptides with overlapping sequences to try and get more information about where the deuterium change is. So by looking at those overlapping peptides, for example here, more information about the exchange can be obtained. So you can see that in the shorter peptide, there's slightly higher uptake than in the longer peptide, suggesting that there is a difference overall from the N-terminal to the C-terminal part of the peptide. In this example, the authors used deuterium exchange to identify the site of antigen binding on an antibody. So here, the antigen was bound by the monoclonal antibody, and you can see protected regions when the antigen bound are highlighted in red. And these were identified based on changes in the deuterium uptake. So this is just an example of how one can use the process to map changes in protein conformation and try and identify where ligands are binding, where effects are taking place. So for more information about these types of methods and to think about how you could use them in your own research, please feel free to contact your local mass spectrometrist. Within the NCI, you can talk to me, Lisa Jenkins, or my colleague Thorkel Anderson in Frederick. Within the wider NIH community, I would direct you to visit the CREX for more information about available resources. Thank you very much for your attention.
U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4CANCER. Produced August 2019.